So let's go through question 1 of BLAST Day 2. I'll share the screen with you. So this is the Day 2 BLAST that we're doing homology-based gene prediction. All right, so here I'm just reminding you what genes are, since we're using BLAST to do gene prediction. And what I want is to say, okay, given a genomic sequence which has genes, maybe, and non-genes, maybe. Most of the time it's a mixture of both when you think genomic. And if you think prokaryotic, then mostly genes. If it's eukaryotic, then we would guess that it's mostly non-genes. So given a genomic sequence, are there genes in that sequence that we just obtained? So actually at this, guys, we don't run it. I'm going to run question 3. I said I was going to do question 1, but here's question 3. So when I'm asking how many genes are encoded in the human genome, not the whole human genome, just this chunk of it. So what you do is you copy that, copy the sequence, and I want to know, given this sequence that right now I have no idea, does it encode anything or not? Then I want to delete that. Okay, and right now I'm running BLAST, nucleotide BLAST, and I'm going to run, as my first run, I'm going to do it with the nucleotide database. So it's going to see what does this sequence match. And you might wonder, but why are you doing that? And you will see when I run, you'll see in the results. Right now, all I'm asking is, what does this whole sequence that I put here, what does it match when I compare it to the database? And you've done this before when you're trying to figure out, maybe you don't even know to which organism this belongs. And what you're doing is identifying it. So what we're doing now is, let's see if the results give me anything about the identity of the sequence in terms of, does it contain genes or not? So then you run that. And while this one is running, I'm going to go on this next screen. Oh, I guess that one was pretty fast. So let's just see what the results are. Okay, so we got lucky. Okay, I already knew it was from humans, but if I didn't know, then it would say most likely it's humans. That's the best match. And I know it's for a gene. So we got really lucky. So yes, there is a gene encoded there. And I even know what the gene does. And so there are other candidates too, which apparently are the same gene. And this has some domain of E3 ubiquitin, which is possible to exist within that gene. I don't know enough about this specific gene to know if it does have this domain. And let's see, so this, on the top are the best hits, right? So, and then mRNA, you're like, oh, okay, there's an mRNA sequence that matches that. That's more evidence that that's a gene. cDNA, that's complementary DNA. That's also evidence that that DNA sequence gets transcribed. cDNA is a DNA molecule that is made by having the mRNA. You have the mRNA molecule, and they use an enzyme to make a second strand that is complementary to that mRNA, and that's the cDNA. So people do that just because of technical difficulty is that mRNA degrades pretty easily. So you want to work with molecules that are DNA, not RNA, because RNA is hard to work with. As you probably heard about the vaccine that is RNA vaccine, how hard it is to transport, to keep, or whatever. So let's see. Yeah. And this guy here just says which chromosome does it belong to. It doesn't tell you, is it a gene? Is it transcribed? Is it translated? What is it? Oh, I didn't mean to click on that. Go back. Okay, well, it's not going back, so I'm going to go back from the top. So I was here on descriptions. The other place where I can look where the matches are is here on graphic summary. And the top one is saying, basically my whole query, that sequence I entered is this thing, which is, if I hover over, it says, is that a gene that does the best match? So really what I entered was the sequence for that gene. And I said it was genomic, which was genes plus non-genes. Well, it looks like I lied. It was, I only entered gene sequence. And then there are other matches. Let's see, oh, the gene also. And these might be like different species. Let's see. Predicted. Okay, the predicted is less trustworthy, which is like, it seems like this would be true, but we don't have lab information to say it is true. So this is from bioinformatics. Whenever something says predicted, so let's go to the results. This guy's here. It's not that it's not true, it's just that this is based on sequence similarity, but somebody has not necessarily isolated that mRNA sequence from this. All right, so when I'm asking how many genes are predicted in that sequence, it's only one, because it's the one bar, no interruptions, that's that one gene.